Chapter 6 is on microbial growth, and this is part A of this chapter. We're going to talk about the requirements for growth. We're going to look at temperature and pH and osmotic pressure and see how these influence the bacterial cell. For microbial growth, it's important to remember that for bacteria, they're going to increase in number of cells as a population, but don't necessarily increase in size. Bacteria don't get big and fat the way we talk about humans. When we see that cupcake, we certainly know what to do with it. But for bacteria, growth means how many cells are going to be produced. In order for them to develop into a large population, they have requirements both physical and chemical. For physical, we're going to look at their temperature, optimums, pH, how osmotic pressure influences their growth, and we'll look at the basic chemicals that are needed for growth. We often talk about the acronym CHOMPS, which indicates that carbon and nitrogen, sulfur and phosphorus are going to be major elements, but we'll also see that there's other trace elements and organic growth factors, and again, oxygen requirements are going to vary according to the bacteria, as we saw in Chapter 5, based on their metabolism. So with temperature, we see that some bacteria like it hot and some prefer the cold. So when we look at a temperature growth curve, we're going to see a minimum growth temperature where below that bacteria cannot survive. And at the other end, their maximum growth temperature, which at the highest temperature, the bacteria can no longer survive. At the peak, they will be at their optimum growth. And therefore, the bacteria is liking that temperature and growing as quickly as it can. When we look at the classification of bacteria, we talk about those that are cold-loving, which are psychrophiles, compared to those that may be heat-loving, which are thermophiles. Most of the clinically relevant organisms that we'll talk about are actually going to be mesophiles. These are those in the moderate temperature-loving range, and most of them prefer body temperature. So this diagram looks at what a typical growth curve looks like. You see that the slope is a little bit more gentle, whereas after the peak at the optimum, the curve usually quickly drops off. That's because when bacteria get above their optimum, that excess heat usually is going to kill them more quickly than what cold will. So if you look at the mesophiles, we see that they have a minimum growth temperature at about 10 degrees Celsius. Their maximum is about 50 degrees Celsius. That's why cooking is a very effective way to kill many of the bacteria that we come in contact with. These disease-causing, not everything that's a mesophile is disease-causing, but we see most of our clinical isolates coming from this category. They have an optimum at our body temperature, which is around 37 degrees Celsius. We also see that the psychrophiles, things that can grow below 0 degrees Celsius, are going to be environmental isolates. And as well, the thermophiles and hyperthermophiles, we don't typically see organisms that can survive above 100 degrees Celsius. These hyperthermophiles are often found in places like very hot deep sea vents and such. So we're going to focus mostly on the mesophiles, but we'll also talk a little bit about the psychrotrophs. The psychrotrophs are able to survive in refrigeration temperatures. As you can see, their minimum goes down to 0, but they actually are going to have an optimum that is about 22 degrees Celsius, and so therefore they go very easily from the environment into the refrigerator and are often involved in food spoilage. So just looking at those categories, we see, again, this special group of psychrotrophs that aren't necessarily outright pathogens, but are very important in food spoilage because they do survive refrigeration. Psychrophiles, they're unique because they can grow below 0. However, they are going to be unusual environmental isolates. The same with the thermophiles and hyperthermophiles. So we're really going to focus on the mesophiles and the ability of these organisms to cause disease. We can also talk about, though, in general, the interesting relationship between microbes and food spoilage. So microorganisms, as you know, are in all of our food. There can be bacteria, fungi, even viruses. Some bacteria could be pathogenic, so we'll talk about things like Listeria monocytogenes and its issues. But some of the bacteria just are involved in spoilage. The same can be true of the fungi and yeast. The relationship that we see between food spoilage and nutritional status, though, is quite problematic when we think from the standpoint of nutrition and its relationship to what we're learning about the microbes. Unfortunately, food that has the highest nutritional value, things like vegetables and our sprouts and fruits, these are the things that have the fastest spoilage rate. Bacteria and fungi grow very quickly on these foods, and that's problematic. If you're on a limited budget, having various fruits and vegetables that are fresh, it's problematic because you can't afford for those things to go bad and just be thrown out. So if we look at the other end of the curve, we see things that are the lowest nutritional value are the things that are the slowest to spoil. That's why we see things like processed junk food, that they have a very long shelf life. And so that's why these types of foods are problematic when we have people in poor socioeconomic areas. They often are in what are referred to as food deserts because they can't get access to fresh fruits and vegetables. The exception might be things like dehydrated fruits and veggies and nuts and things, but in general, we see this unfortunate correlation that for most foods, the faster it spoils, the higher the nutritional density, but it's spoiling because the microbes like all the food that's there as well. We also see that when we're handling our food, not only do we need to think about the psychrotrophs that are the food spoilage organisms, so as we said before, they are the ones that are going to survive refrigeration. So refrigerator temperatures are meant to be about 0 to 5 degrees, and that usually is going to inhibit most true pathogens. So very few of the true pathogens actually grow in the refrigerator. However, all refrigeration is doing is slowing the growth of bacteria. And so therefore it gives us a longer shelf life for those foods, but it's really only slowing the growth of the spoilage of those foods by the bacteria and other microorganisms because they will continue to grow. And I'm sure you do that experiment a lot in your refrigerator when all of a sudden you find something in that Tupperware container that has gone bad. When we talk about our mesophiles, though, we're concerned about them when they're in the danger zone. So, yeah, they have their optimum at 37 degrees Celsius, but they're going to grow fairly rapidly between about 15 degrees Celsius up to about 50 degrees Celsius. That rapid growth of bacteria means that many of them are able to produce toxins in that danger zone, and that is part of what makes them problematic pathogens. So we need to make sure that we're holding food at appropriate temperatures so that we don't have things producing toxins in that danger zone, nor do we want things spoiling by refrigerate. Even though they're growing in the refrigerator, they still may not be outright pathogens, they can still potentially make us sick. When we think about how we handle our food, one of the problems we always see is when we try to do large amounts, whether it's cooking large amounts or cooling large amounts. Here we see the effect of the amount of food on cooling rates in a refrigerator and its chance of spoilage. So one bacteria that's associated with rice is Bacillus cereus. Bacillus cereus is common out in the environment, on the handprint plates that we saw in the microbial safari, maybe you notice some of these kind of irregular colonies that are picked up as transient organisms. So this is Bacillus cereus, a gram-positive rod that forms endospores. Well, Bacillus cereus is a foodborne pathogen that can produce toxins. They can produce either what is referred to as the emetic type of toxin, which causes vomiting, or you can have the diarrheal syndrome. The emetic toxin is the cerulide, which is produced when the food is usually at the wrong temperature, and therefore the Bacillus cereus will grow and produce these types of toxins that, again, can be problematic. And therefore, proper food handling is especially important after cooking to prevent these illnesses. Getting things chilled to the appropriate temperature so that the bacteria can't grow and produce toxins are important. So we see in the diagram here that the dish on the left-hand side, represented by the blue line, it's nice and shallow, so it's got good surface area to the cold. So as we've cooked our rice and we then cool it in the refrigerator, we see that the temperature is going to drop very quickly so that the food gets down to refrigerated temperature probably in under an hour. That doesn't give a whole lot of time for the bacteria to be able to replicate and produce the toxin. On the other hand, when we tried to supersize this, we needed to make a large pot of rice. We see that our dishes are often deeper because of the size of the burners on stoves and things. We don't end up elongating things, but they go in more depth. And so you're going to have a core that it's going to be more difficult to heat or cool, depending on if you're cooking or cooling afterwards. So the red line represents this deeper dish. After we have cooked the rice, the inner core is going to stay in the danger zone for a significant amount of time. You see the danger zone there in the darker color. The red line is going to mean that the bacteria is actively growing during those several hours and producing toxin, which could potentially make people sick. So it's important to be thinking about these types of physical requirements. Besides temperature, we also see pH is important for the growth of microbes. Most bacteria grow best between a more neutral pH, between pH 6.5 and 7.5. Molds and yeast, on the other hand, prefer more acidic pH at around 5 to 6. We again see we can classify our microbes based on what type of environment they like according to their pH. Things that are acidophiles are those that are acid-loving. They can grow in very acidic environments. There are some extreme organisms, because we have a lot of microbial diversity out there. The chemoautotrophs can actually survive in the coal mine drainage. And these organisms, they're able to oxidize sulfur to sulfuric acid. That means that they can survive in something that's around a pH of 1. It's really hard to get anything down to that low of a pH, so this is basically pure sulfuric acid that you would think would just kill everything, yet we have some of these extreme bacteria that are able to survive. In the lab, when we have culture media, whether it's in liquid form or whether it's in agar plates, we have to incorporate things like peptones and amino acids and phosphate salts. These, not only are they nutritional value, but they also act as buffers in the media so that you don't have pH swings that would be detrimental to the growth of the organisms. So if we look at some interesting things with pH, we look at the scenario of what happens with milk as you have microbial succession. So initially, even though we pasteurize milk, it doesn't mean that it's sterile. There are still organisms there, and that's why milk goes bad. So when we start out with high populations, we have organisms initially such as Streptococcus and Lactobacillus. If you remember, these are organisms that can ferment various carbohydrates. So they're going to drop the pH, and as we see on the right-hand scale, these may drop the pH down to around 4, and that's why we then see the spike in growth of things like yeast and molds, which like that more acidic environment. We see these as well may then contribute to a drop in pH, and then we see another unusual organism, our bacillus, this gram-positive rod, showing up at that point. So the pH is going to be important, and it's going to determine which bacteria are going to be most viable at that particular pH. We can also talk about physical requirements from the standpoint of osmotic pressure. We looked at osmotic pressure in chapter 4 when we talked about the ability of the cell wall to help protect from osmotic lysis and things that get across the membrane. Well, osmotic pressure is important. Most organisms, because they are 80 to 90% water, are going to be affected by both hypo and hypertonic environments. So in a hypertonic environment, when we increase things like salts or sugar, remember that causes plasmolysis. The cell is going to dehydrate, so as we add these salts or other solutes, sugars and things, that's going to pull the water out of the cell, and the cell is going to no longer be metabolically active. So this is going to be a great way to try to preserve food. Bacteria, though, have developed a lot of diversity in their ability to be able to grow. So every time we talk about these types of requirements, there's always the exception to the rule. Here we see that some bacteria are halophiles. Halophiles means salt-loving, and we have both extreme or obligate halophiles that require an extremely high osmotic pressure. These are only found out in the environment, and so a great example is the Dead Sea. Bacteria that grow in the Dead Sea may actually require up to 30% salt. If you remember, physiological saline is around 0.9%, so this is just an extreme amount of salt. And the reason people called it the Dead Sea is that very few things can survive there, but it's not devoid of microbial growth. Facultative halophiles, on the other hand, are able to tolerate high osmotic pressure, high salt, but not quite as extreme as those other obligate halophiles found in the Dead Sea. So things like Staphylococcus, Staphylococcus aureus, Staphylococcus epidermidis, these are usually able to grow at things that are above 2% salt. And in the case of Staphylococci, they actually can survive maybe even up to 15% salt. And so therefore, Staphylococcus is going to be a major clinical isolate that we are going to be talking about as a facultative halophile. Here you see just a reminder of what it means for a cell to be plasmolyzed. So as the cell in figure B there is placed in 10% sodium chloride, you can quickly see that the water is going to be drawn out of the cell, and the plasma membrane is going to shrink away from the cell wall, and that is referred to as plasmolysis. So that's why primitive civilizations use many of these types of techniques relying on osmotic pressure to preserve food. Meat would normally just spoil and spoil very quickly. So preserving it with salt meant that you could have meat stay around for a longer period of time. The same way that when we talk about things like preserves, well, that was the problem. When you had fresh fruit, it is going to spoil very quickly. If you preserve it by putting high sugar content, you are going to be able to pull the water out of the cells, and therefore the bacteria are not going to be able to carry out metabolism and grow. So in essence, you're preserving these types of foods. So this was a great way to be able to store things for more long term at a time before refrigeration. Besides these requirements for the physical environment around them, we're also going to talk about some of the things that the cell needs for growth, such as elements, carbon, nitrogen, sulfur, and phosphorus, and also look at their ability to utilize oxygen. When we talk about the chemical requirements, bacteria, like us, are carbon-based life forms, so it's not surprising that if you dried down the cell and took out all the water, carbon is about 50% of the dry weight. It's the structural backbone for all living matter, and so it's our enzymes, it's our structural proteins, and as chemoheterotrophs, most of the organisms that we're talking about are using organic carbon sources. Nitrogen, on the other hand, is about 14% dry weight. That's still pretty high, and if you think about the fact that cells are mostly proteins, then again that makes sense because nitrogen is found in proteins and the building blocks of proteins, amino acids. Most bacteria can easily decompose proteins, and that's why they are a major decomposer of anything that is a protein-based material. Some bacteria even use nitrogen in the form of nitrates and ammonias and things, and a few bacteria out in the environment are very important in nitrogen fixation. So we see that many of the soil bacteria are involved in nitrogen fixing and denitrification, and so these are just part of the natural cycle of nitrogen. Sulfur and phosphorus are about 4% of the dry weight of the cell together. And that's a little bit surprising if you think about it. So sulfur is found in amino acids and things like thymine and biotin, and most bacteria, because they decompose proteins, can easily get sulfur. Some bacteria even use sulfur in the form of things like sulfates or hydrogen sulfide, but they're basically going to use that sulfur to be able to make amino acids as they need to build proteins. What's surprising is that phosphorus is not needed in higher amounts. If you think about phosphorus is needed to make DNA, RNA, ATP, all of our membranes. So it's kind of unusual that there's not more of a requirement for phosphorus, but it is what it is. Phosphates are usually the source of phosphorus for the cell. And similar to us, bacterial cells need other elements. So we take our vitamins and we need to get our calcium and magnesium and potassium. Well, so do bacteria. They usually get that in various organic compounds to be able to provide them with these essential elements. The other thing that we don't think about, but are still equally important, are the trace elements. Trace elements are inorganic elements that are required but only in very small amounts. These are usually enzyme cofactors. So when we talked about the electron transport chain in Chapter 5, we saw that many of the cytochromes are using iron as that moiety that undergoes oxidation and reduction for the cytochromes to be able to play past the electrons. So iron is an important example of a trace element. And we know if we or bacteria don't have enough iron, you're just not going to be able to produce the ATP because iron is so important in that electron transport chain, as we saw. But other trace elements that we need are things like copper, molybdenum, and zinc, and these can be provided in very small amounts, but they are essential. Other organic growth factors usually come from the organic compounds that are obtained from the environment that the microbe finds itself in. So it gets vitamins, amino acids, and other things from the environment. So it can take in larger macromolecules and utilize them for the purines and pyrimidines and various other organic growth factors. The other requirement that you may not think about is the need for oxygen. When we were talking about aerobic and anaerobic respiration, we correlated that in Chapter 5, with the amount of ATP that an organism can make. So many organisms require oxygen to carry out aerobic cellular respiration. In this case, they're going to use hydrogen atoms that combine with oxygen as the final electron acceptor to form water as the last step in that cellular aerobic respiration, and that yields a great deal of energy. For microbes that use molecular oxygen, those aerobes, the bacteria that are aerobes are able to get 38 ATP for every 1 molecule of glucose. However, we didn't talk about it as much, but anaerobic respiration also is going to be a moderate energy yield. So with the anaerobes, we saw that that was always going to be greater than 2, so more ATP than fermentation, but it couldn't give as much ATP as what we saw in aerobic cellular respiration. Thus for anaerobes, they can only make between 2 and 38 ATP. We don't have a lot of examples of anaerobes that we're going to be talking about this term, but one classic example is Clostridium. Clostridium tetani, the causative agent of tetanus, Clostridium botulinum, the causative agent of botulism. Those are going to be our go-to anaerobes. So for the oxygen requirement, we can talk about this from the standpoint that our aerobes, they're going to be able to produce that 38 ATP, so that's why they're doing their jumping jacks and sit-ups as the cartoon indicates, but those anaerobes are just a little bit more sluggish because they can't use all the intermediates in the Krebs cycle to produce as much ATP. So this requirement for oxygen, we can measure this in the lab by using what is referred to as a reducing media. The reducing media that is being pictured here is thioglycolate, that this is a broth that we can put into a tube, and look where the bacteria are going to grow. In a reducing media, it binds up the oxygen, so it's not necessarily available readily for the bacteria farther down in the tube. So we see that the oxygen is going to be highest at the liquid line, and as you get farther down into the bottom of the tube, oxygen is not going to be present. Therefore, if we have obligate aerobes that can only grow by aerobic cellular respiration, they're only going to be able to grow at the top of the liquid where there's the most oxygen present. In contrast, obligate anaerobes that are killed by oxygen, they're only going to be able to grow at the very bottom of the tube. They can't utilize the oxygen and they're actually harmed by the oxygen, and so they will grow at the bottom where the oxygen is not being made available. We do have some bacteria that are switch hitters. Facultative anaerobes are ones that are going to prefer where the oxygen is. They grow better at the liquid line. However, they still can carry out forms of fermentation to be able to grow farther down in the tube as well, where the oxygen is not found. Aerotolerant anaerobes, on the other hand, are going to grow evenly throughout. They don't care where the oxygen is, it doesn't matter, so they just grow wherever, and it doesn't matter what the oxygen level is. Microaerophiles, on the other hand, usually grow in a very distinct band, a little bit below the liquid line. These organisms have more adapted to their environment, which is usually CO2 enriched. So in places like the throat or your GI tract where there's just not as much oxygen available, so they could only handle oxygen in small amounts. Why would that be true? Well, one of the weird things about when we started to have a oxygen-based world, it allowed eukaryotes to become multicellular and more energy to be produced by things like aerobic cellular respiration, whether for bacteria or a eukaryote. However, the dark side is that oxygen can also be toxic. You know that you take antioxidants in your diet, so you're trying to eat your broccoli and things because they are rich in these antioxidants that are going to help bind up these harmful forms of oxygen. Oxygen becomes problematic when it's boosted to a higher energy state. Usually what happens, it is able to then damage DNA, and in a eukaryotic cell, that can cause anything from cancer or other type of abnormalities based on damage to the DNA. For a bacteria, when this toxic form of oxygen damages its DNA, it usually just dies. So unless the bacteria can figure out how to detoxify that oxygen, it's going to have problems with these types of oxygen states. So the worst of the worst is what's referred to as the superoxide free radicals. These are highly reactive and very damaging to DNA. And so we see that bacteria, and actually our cells as well, have an enzyme, superoxide dismutase. Superoxide dismutase is universal in all cells, and except for those that are anaerobes. And we see that this is going to allow the worst of the free radical to be converted to a less harmful form, hydrogen peroxide. Hydrogen peroxide, however, still is somewhat toxic. So in addition, there are enzymes such as catalase and peroxidase that are going to convert hydrogen peroxide, H2O2, to water in the case of peroxidase, and water and oxygen in the case of catalase. So that's why when you put hydrogen peroxide on a cut, you may start to see bubbles, not only from the bacteria, but also your tissue catalases as well. So it's important that we have, and bacterial cells have, these enzymes. But it's these enzymes that really dictate where the bacteria are going to be able to grow. So if we look at obligate aerobes that are only able to grow at the surface where the highest level of oxygen is, we see that they can grow there because they have those enzymes. They have the enzymes catalase and the superoxide dismutase, SOD, that allows them to detoxify the oxygen so that they're not harmed by those types of radical oxygen species. On the other hand, obligate anaerobes, they cannot survive in oxygen. That's because they lack the enzymes to be able to detoxify. So they do not have SOD, they do not have catalase, they don't have peroxidases, and that's why they can't tolerate the oxygen. Oxygen kills them because those radical forms start to damage their DNA. So they can only grow in the thioglycolate at the very bottom where the oxygen has been removed. Our switch hitters of the microbial world, the facultative anaerobes, when they can, they're going to use aerobic respiration. That's why, because for every molecule of glucose, they get 38 ATP. So they prefer that type of metabolism when they have oxygen present, and they will be able to grow quicker. That's why there will be more growth at the top of the liquid line where the most oxygen is, because they will carry out aerobic cellular respiration there. However, when oxygen is not available further down in the tube, they're going to be able to switch over to fermentation. So these organisms grow best where the most oxygen is present, but can grow in diminished amounts throughout the tube. This is, again, similar to the obligate aerobes. They have those enzymes, catalase and the superoxide dismutase, the SOD, that allows them to detoxify those harmful oxygen radicals. In D, our aerotolerant anaerobes, they don't care. They have growth that is evenly throughout. They have one enzyme, only the SOD, the superoxide dismutase, that allows them to take care of the most harmful form of oxygen. So they basically can only partially neutralize. So therefore, they're just tolerating the oxygen, but they just don't do very well. And that's why they grow evenly throughout. The microaerophiles, as we said before, are going to be in a very concentrated band. However, they can detoxify oxygen, but because they are normally found in areas like the throat and GI tract where oxygen is not plentiful, they really don't do well in the large amounts of oxygen. That's because they're just not used to being exposed to atmospheric oxygen. So in order for us to culture anaerobic organisms, or those that prefer CO2, we can either use things like reducing media, as the thioglycolate is, which contains chemicals that combine with the oxygen. We can heat off the oxygen, and that way it is able to allow us to grow both aerobes and anaerobes and figure out their oxygen requirement. When we put things on plates, though, we have to try to provide that environment. So if we have things that we have isolated from the throat, a throat culture, and put that on a plate, we usually need to provide those organisms a CO2-enriched environment in order to be able to grow. So in modern microbiology, we can do this by having these little packets that contain things like sodium bicarbonate and sodium borohydride, that when we add water, this is going to cause the production of CO2. That allows us to grow these organisms from the respiratory tract or even the GI tract that are referred to as capnophiles, and that they are CO2 loving. In the olden days, we used to use things that are referred to as a candle jar, and that's literally what they were. You took kind of these big glass pickle jars and put your plates in there and put a candle on top, lit the candle, sealed the lid, and the candle was going to burn as long as there was oxygen. Once the oxygen was removed, the candle went out, and we then provided a nice anaerobic environment, or at least a CO2-enriched environment, for those bacteria to grow. If you truly want to work with obligate anaerobes, though, that are actually killed by atmospheric oxygen, you have to work in a specialized anaerobic chamber, what's often referred to as a glove box. So it's this chamber where you need to put all your materials in there, and then you remove all the oxygen. Obviously, people can't be in that environment, so you're outside this box, and you manipulate the media and bacteria by putting your hands through these gloves, as seen here. Hence, it was referred to as a glove box. So this is the only way that you can truly work with obligate anaerobes, because they would simply die if they were exposed to atmospheric oxygen. So we've looked at the need for bacterial cells from the standpoint of their chemicals and their oxygen requirement. And we see that the obvious way for a bacteria to avoid the problems with oxygen is to detoxify it. It can't always control what environment it finds itself in, but for those organisms that are able to use things like sod and catalase, they're going to be able to survive in a variety of environments because they will detoxify those harmful oxygen-free radicals. The other thing about growth of bacteria is that sometimes they like to congregate in what's referred to as a biofilm. So bacteria originally are in a planktonic form, meaning it's just a single bacterium, but they usually attach to surfaces. Once they do, they start to create a jelly matrix, and these bacteria then start to form these microcolonies in multiple layers. Once this biofilm matures, they form this hydrogel that looks like this mushroom body. This hydrogel is made of polysaccharides that really protect the bacterial community, and this can be problematic when we talk about these biofilms from a clinical standpoint. So let's take a look at these biofilms. Interestingly, about 70%, maybe even more, of human bacterial infections actually probably are caused by biofilms. These biofilms are these functional microbial communities. The microbes adhere to surfaces and then accumulate. Once they do, they go through what's referred to as quorum sensing. They know that their neighbor is there because they send out chemical signals, and they want to protect the community. So they start to make this hydrogel, which is a slime layer. But the problem is that hydrogel is very difficult for things like antibiotic and disinfectants to be able to penetrate. Once things start to grow in these microbial communities, they may become more than 1,000 times more resistant to antibiotics. So we see these types of biofilms all the time. When we wake up in the morning and we have that slimy layer on our teeth, that's a biofilm. People that wear contact lenses, if you have ones that are not the disposable, you have to constantly clean them because you start to get this type of biofilm on them. Notoriously, catheters, permanent lines, implants, most of these fail because of infection. The infection isn't just any ordinary infection. It usually comes about because of these biofilms. We can't just simply throw 1,000 times more antibiotics or disinfectants at things, so we really need to start coming up with novel ways to treat these biofilm infections. Antimicrobial peptides and antibiofilm drugs are kind of a hot area of research. There's lots of opportunities to try to think about preventing the formation of biofilms, but there's probably more challenges than there are solutions at the moment. However, one interesting approach is to use lactoferrin to try to prevent the accumulation of these microbial communities in the biofilms. Lactoferrin is going to bind iron. Bacteria, in order to grow, they need iron. It's part of that electron transport chain. So if you give lactoferrin and prevent the iron from being available to the bacteria, you can inhibit their growth and thus inhibit the biofilm formation. Here we see what that biofilm might look like. You see this kind of mushroom community, so that hydrogel with all the bacteria inside. But interestingly, that mushroom shape is what's important for allowing water and nutrients to be able to still get to the community. Biofilms are problematic, and in a real-world example here, we see a scenario where patients that have indwelling catheters received, unfortunately, contaminated heparin. However, when we did counts on the bacterial numbers in that contaminated heparin, they were too low to really cause infection. However, 84 to 421 days after exposure, a number of these patients developed infections. Why? Because of these biofilms. So we see that primary sites for biofilm infection are things like the places where those permanent lines and other catheters are going to be put in. So we see an example of the subvenous catheter spot where things are going to be used for an inline portal. The mouth is a primary. Artificial hip implants, unfortunately, become a major issue for biofilm formation. But once those biofilms form, you saw in the previous picture, some of those clusters can break off. And then we see they travel to other areas and cause secondary biofilm infections. So places like the liver and kidney and brain. And so understanding and treating biofilms has become a major issue in clinical infectious diseases. So understanding biofilms is important because this is a major virulence factor for those bacteria that form biofilms. They make that hydrogel so they're protected from antibiotics getting in, from disinfectants from getting in, and so it's a great way for them to be able to grow and proliferate without being able to be killed by our normal forms of treatment and disinfection. Now we're going to talk about culture media. We're going to see how we actually grow bacteria on artificial augers. In the lab, unfortunately, this term we're not going to be able to see a lot of this, but we can at least talk about it. So we can utilize what is referred to as complex medias. The brain-heart infusion agar that we use for our bathroom microbial safari is an example of a complex media. You take nutrients from leftover animal parts at the slaughterhouse, brains and hearts and things, and it has all the nutrients and vitamins and carbohydrates that's needed, but you don't know in what amounts. In contrast, we can also make chemically defined media in the lab. We could measure the amount of protein, the amount of carbohydrate, and all the other nutrients so that we know what's exactly is there. So both of those can be useful. Oftentimes, though, if we're just trying to get things to grow, we're going to use a complex media. Clinically, though, we often use selective and differential medias. These help us to actually identify and isolate pathogens of interest. And unfortunately, as we take bacteria out of the body, oftentimes these can be hard to grow, so enriched medias are important as well. So in selective medias, they contain chemicals to inhibit the growth of one type of bacteria while permitting the other to grow. So we have some agars where they may be selective so that only gram-positives may be able to grow. These selective medias usually don't have indicators. They're just kind of a all or nothing, something grows or not. Differential medias contain chemicals that allow differentiation of those bacteria that are growing. So they may be closely related, but we're trying to find ways to figure out a specific species. So these types of medias usually do have indicators that are going to allow us to identify one closely related bacteria group from another. So some of the common selective medias that we use in the lab are things like PEA. PEA is phenylethyl alcohol agar, and this one selects for gram-positive organisms. So things like Bacillus, Staphylococcus, Streptococcus, but it's going to kill gram-negative organisms, things like E. coli, Enterobacter, Salmonella, Shigella. If you think about it, this is the same principle that we talked about with the gram stain. When we put the alcohol on for the differential step in the gram stain, that alcohol dissolved the LPS layer, and therefore the cell wall, being very thin in gram-negative organisms, started to fall apart, and the cell got leaky so that it was easily destained. However, in the real world, that cell is probably going to die once that LPS has been removed and its cell wall starts to fragment. And so the PEA agar, because of the alcohol, kills those gram-negatives, leaving the gram-positives to survive. In contrast, crystal violet is a basic dye. So if you remember the chart from chapter 4, we saw basic dyes in general are going to kill gram-positives. Because they have the thick peptidoglycan layer, it's very porous and allows these small dyes to get in. However, because gram-negatives have that LPS layer, it's a little bit harder for these dyes to get in, so crystal violet and other basic dyes kill gram-positive organisms, but allow the gram-negatives to live. As we saw before, the principle of osmotic pressure, salt agar, if we have agars that contain a lot of salt, 7.5% sodium chloride, compared to physiological saline, which is 0.85% sodium chloride, this is going to select for halophiles, those salt-loving organisms. So if we put these together, we often utilize the two as one media. So selective and differential medias are very important. We're going to take a look at some commonly used selective and differential medias and talk about things like mannitol salt agar, MSA, which the salt is the selective agent and selects for halophiles, but then we can incorporate a carbohydrate, in this case mannitol, that is going to allow us to differentiate organisms into mannitol positive or mannitol negative organisms. The mannitol salt agar is kind of this orangish, reddish color. MacConkey's agar, on the other hand, is going to select 4 gram-negatives. These are going to be organisms, again, that the crystal violet is going to kill off the gram-positive, so the gram-negatives are going to survive. Of the gram-negatives that survive, we're then going to use another carbohydrate, in this case lactose, to differentiate things into lactose positive or lactose negative. So MacConkey's looks a little bit like the mannitol salt, but you can tell that it's a little bit more purplish because of the crystal violet. And then eosin methylene blue, or EMB, is the same principle as the MacConkey agar, but instead of crystal violet as the basic dye, it has 2 other basic dyes, which are eosin and methylene blue. So again, this is going to select for gram-negatives, and it as well uses lactose as the differential agent. So if we look at mannitol salt agar, the halophile that is most relevant clinically is Staphylococcus. So this is going to select for halophiles in general, but usually what we're actually looking for are the various staphylococci that are of clinical interest. So mannitol salt agar contains 7.5% sodium chloride to inhibit the non-halophiles, allowing the halophiles to grow. And then we use the mannitol to differentiate into mannitol positive and mannitol negative. This is important because if we look at the genus Staphylococcus, we have things like Staphylococcus epidermidis, which is mannitol negative, and it's just normal skin flora. But Staphylococcus aureus is a major, what is referred to as an opportunistic pathogen. It's very important that whenever we find Staph aureus, we can readily identify it. Well, Staph aureus is going to be mannitol positive. So what that means is that organisms that can ferment the mannitol, that's just another carbohydrate, the end product is going to be various acids, and that's going to lower the pH. And because it has the pH indicator, phenol red, incorporated in the media, the agar will now turn yellow if you have a mannitol positive organism. So when we look at Staph epidermidis and Staph aureus on the plate, we can differentiate them. Both of them will grow because they are halophiles, but on the left-hand side, we see Staph aureus has this nice yellow halo around it, indicating that it has fermented the mannitol and produced the acid, changing the color of the media to yellow. On the right-hand side, we see Staph epidermidis is growing, but it can't ferment the mannitol. So being mannitol negative, there's no change to the color of the media. Up above, whatever organism was streaked there, it's a non-halophile. There's no growth because it couldn't survive the salt. MacConkey's agar is going to be used for differentiating enteric gram-negative bacteria. So these are bacteria usually found in the GI tract, and you really need selective and differential media here because there's so many organisms that are found in fecal material. So the crystal violet is going to inhibit gram-positives. We're not looking for them right now, so we're interested in selecting for the gram-negatives. So we have gram-negatives growing on the plate. Now we're going to differentiate them based on their lactose fermentation. So lactose-positive organisms are again going to ferment the lactose and cause a drop in pH. And in this case, the color is going to turn the organism pink. These types of lactose fermenting bacteria are referred to as coliform bacilli. So these are gram-negative rods that are associated with fecal material. That's why you'll often hear people talking about doing coliform counts to determine if fecal contamination is present. So these lactose positives are normal fecal flora, so things like E. coli and Enterobacter. However, lactose negative organisms are not going to be able to ferment the lactose and are going to be uncolored on the agar. These are potential pathogens such as Salmonella and Shigella, which cause things like dysentery and typhoid, and so definitely we want to identify these potential pathogens in a fecal sample. So on MacConkey's agar, we see the typical reactions here. On the left-hand side of the plate, we see Salmonella is colorless. So it's hard to see, but it's the kind of orangish squiggly line that's just the color of the agar being colorless there. That is lactose negative. The middle one is Enterobacter. So this has turned the organism actually pink because it's fermenting. The lactose positive one is E. coli. E. coli produces so much acid, it usually causes a precipitate or a cloud around the colonies, or in this case, the streak. EMB, or eosin methylene blue, is the same principle. So the eosin and methylene blue are the selective agents that are going to inhibit the gram-positives. They're just basic dyes. And then we're going to look for, again, those organisms that can ferment lactose. So it's going to differentiate the gram-negatives that are on there. So in this case, on this kind of brownish purplish agar, E. coli, which is positive, is going to be a dark purple. But again, because it produces so much acid, it also gets this really pretty green metallic sheen. Other coliform bacteria are going to be things like our Enterobacter. So this is going to be lactose positive, but be more of a purple pink color as we see these as normal fecal flora. The lactose negative ones, again, will be colorless. So our potential pathogens that we're usually screening for, Salmonella and Shigella, and we'll continue to talk about those in our GI chapter later in the term. So looking at the EMB agar, we see to the left-hand side, the Salmonella is lactose negative, and so this one is going to just be kind of a light color, but really just reflecting the color of the media. The middle squiggle there is our Enterobacter, which is lactose positive. On here, it's kind of a darkish purple color, but we see that the right-hand side, that is E. coli, that dark green. It's dark purple, but then it has that green metallic sheen to it, identifying it. And you can see that in high concentrations in the first couple of quadrants when we streak the E. coli on EMB. Some organisms are fastidious. That just means they're hard to grow. So they require enriched media that provides them extra nutrients or enriched. Blood is often added to enrich things like BHI or other complex medias. And it can also be used differentially because some bacteria make enzymes, homolysins, that lyse red blood cells and cause hemolysis. There are different types of hemolysis. Beta is complete, alpha is partial, and gamma is no hemolysis. So if we look at these organisms on the blood agar, we can see these hemolytic reactions. Some organisms that produce these homolysins and lyse the red blood cells are going to be able to completely cause clearing, as we see for the beta. For the alpha, it's incomplete, and you end up getting this kind of green halo around it as things like hemoglobin and that discolors the agar. The gamma has no hemolysis. These types of hemolytic reactions are often very useful in identifying and differentiating Staphylococci and Streptococci. Again, especially the Streptococci are a little bit finicky to grow, so that's why we always are putting the throat cultures, which has a lot of Streptococci, typically on blood agar. So indeed, in examples here, the blood agar, we see that Streptococcus pyogenes, the causative agent of strep throat, is beta hemolytic. So many of the other Streptococci are either gamma hemolytic or alpha, and therefore looking for the potential pathogen, we would look for beta hemolytic colonies and try to see if Streptococcus pyogenes is present. In the middle plate, we see an example of alpha hemolysis. E. coli has partially degraded the red blood cells, and it gets a kind of green, ugly color there. Staph epidermidis, on the other hand, is gamma hemolytic. it doesn't do anything to the blood agar, and so it grows because this is an enriched media, so just about everything will grow, but there's no hemolytic reaction. So that's why chemically defined medias and the various components to make selective and differential medias are so important. We've learned a lot about how to culture bacteria, and we've also figured out ways to help us clinically to be able to quickly identify our potential pathogens. In Part B, we'll talk more specifically about how bacteria grow and look at their growth curves to try to understand how we control them as well.